Hello, everyone. My name is Isabel Guzman, a current sophomore at Columbia University. This summer, through the APSA Virtual Summer Research Program, I worked with the Bayless Laboratory focused on the effect of branched-chain amino acids on immune system in children with maple syrup urine disease. MSUD is an autosomal recessive disease where the body is unable to process certain amino acids properly. This is due to mutations in the BCKDHA, BCKDHB, and DBT genes, which are all part of the branched-chain alpha ketoacid dehydrogenase complex. This protein complex functions to break down the three branched-chain amino acids known as valine, leucine, and isoleucine, but when it is impaired, there is a buildup of branched-chain amino acids in the blood, causing neurodegeneration in MSUD patients. Many of the children with MSUD who continue to be sick usually tend to suffer from bacterial infections, indicating potential malfunctioning of their immune system. With partnership from CHOP, clinical data was collected to compare the occurrence of infection among the control group, which is CHOP's birth cohort, minus the experimental patients of the study, MSUD, and two known immune deficiency disorders, including 22q and SCID. This data showed a higher prevalence of infection among MSUD patients than the control group. Though the p-value has not yet shown statistical significance due to the small population size of the patients, these results are suggestive of children with MSUD suffering from some immune system defect. In order to study this, there are two particular approaches: genomic editing and pharmacological analysis. One of the few published articles on the effect of branching amino acids metabolism on the immune system focused on studying the first step of branching amino acid breakdown through the BCAP pathway, where branching amino acids are broken down into keto acids as shown in the diagram of the earlier slide. The researchers found that deletion of BCAP1 gene in the cytoplasm resulted in higher leucine levels and mTORC1 activation as well as increased glycolysis. This means that there is research evidence to show that the BCAP pathway promotes CD4 T cell energy and therefore decreases in immune response. This led us to hypothesize that since branching amino acid metabolism affects T cells in the BCAP pathway, it should also affect T cell functioning through the second step, which is the BCKDH pathway directly linked to MSUD. Through the genomic approach, Bayless Lab researchers edit healthy human T cells through Cas9 RNP of the CRISPR system to mimic patient mutations that are essential to immune dysfunction. The pilot experiment involved modeling a knockout BCKDH gene in human T cells and optimizing the system to study the effects of BCKDH gene through its absence. The methods using CRISPR involve purifying the healthy T cells, stimulating them with CD3, CD28, then electroporating with the RNPs using nucleofection. During electroporation, positive and negative regulator guides for T cell proliferation were used, CD5 and CD3D respectively. The remaining guides were used for the experimental group, BCKDH gene, as mentioned earlier. From there, Auto 550 plus and CDT stains were collected, and the editing efficiency was measured through flow cytometry and Whistler blotting. This method is currently being analyzed for final results. In addition to genome editing, another form in which the BCKDH pathway is being studied is through pharmacologically changing branched-chain amino acid metabolism, as shown through clinical studies. We have chosen to specifically focus on BT2, an inhibitor of the BCKDH kinase. The kinase phosphorylates BCKDH, causing it to become inactivated and no longer be able to aid in the continuing breakdown of branched-chain amino acids / keto acids. The BT2 kinase inhibitor then allows for BCKDH complex to be upregulated and therefore metabolism branching amino acids, which can actually then be used as a treatment for metabolic disorders that involve elevated branching amino acids such as MSUD. Although BT2 has potential to help with MSUD, its function to decrease branched-chain amino acids might also have detrimental effects. Referring back to the article mentioned earlier, the conclusions made was that an increase in BCAT1 gene resulted in lower leucine levels, which caused decreased mTORC1, a critical regulator of T cell activation. This means that the BCAT pathway decreases T cell function. Given that BT2 has a similar effect through the BCKDH complex, this inhibitor might also raise complications in CD8 T cell response through the mTORC1 pathway. As shown through CTV stains from patients at CHOP, the BT2 experimental group showed a lower percentage of divided cells. We hope to be able to further explore whether the mTOR pathway plays a role in these results. Understanding the effect of branching amino acid pathways on T cells can help to lead to medical breakthroughs for not only MSUD, but for other metabolic diseases. I would like to thank APSA for this amazing summer opportunity. In addition, thank you to Dr. Bayless and the Bayless Laboratory team, as well as our collaborators at CHOP. It was a pleasure to meet and work with such an amazing team.